Hi, good morning. So here we are about to start our second section, understanding the impact of study design and the role of MIDD on dosage selection and evaluation for combination therapy. So I'm John Liu from Division of Pharmacometrics at the FDA, Office of Clinical Pharmacology. So I'm delighted to moderate this enlightening session along with Dr. Vijay Ivachuri. Where is he? Yeah, he's from University of Maryland School of Pharmacy, and who is also our president-elect of ISoP. So in this session, we have the privilege of hearing from 5 distinguished speakers. So first, Dr. Mark Corway. He's a professor in the Division of Translational Science and Applied Statistics in the Department of Public Health Science at the University of Virginia. And our second speaker is Dean Bodino. He's the Senior Scientific Director and Group Leader for Oncology Quantitative Solutions and Takeda Pharmaceutics. And third speaker is Dr. Donghua Yin. He's the Vice President and Head of Oncology Clinical Pharmacology and Pfizer. And our fourth speaker is Dorothy C. Meand. She is the Senior Director within the Pharmacokinetics, Dynamics, and Metabolism Department and Sanofi. The last one, but not least, is Dr. Aram Organisian. He is the Vice President of Clinical Pharmacology and Non-Clinical Development and Aztecs Pharmaceutics. Additionally, we are honored to have 2 distinguished panel members from the FDA. Dr. Xining Wang, she's the Master Clinical Pharmacology Reviewer in the Division of Cancer Pharmacology 1, Office of Clinical Pharmacology. And Jonathan Valio, he is the Supervisory Mathematical Statistician in Division of Biometrics 9. So you can find more information about their backgrounds on our workshop website. So we will divide the presentation into 2 parts with a 30 minutes break in between. So following the presentation, we will hold a 1-hour panel discussion and a Q&A session. So without further ado, let's start with the first presentation by Dr. Mark Corway from the University of Virginia. So he will discuss statistical trial design considerations for combination therapy. Thank you.
Well, thank you. As you just heard, my name is Mark Conaway. I'm a statistician in the Division of Translational Research and Applied Statistics at the University of Virginia. And I'd like to thank the FDA and ISoP for giving me this opportunity to talk about statistical design considerations for combination trials. As you'll hear through these presentations, as we all know, in embarking on an early phase dose finding trial, there are a lot of decisions that need to be made, including but not limited to what's the starting dose of the agent, what doses you're interested in, what's the definition of a DLT, what do you mean by acceptable toxicity, and if it includes an efficacy endpoint, what is that endpoint, when and how is it measured? When we had an initial discussion about this workshop, implementation seemed to be a key point, and so I took that to heart. And so this presentation is going to focus more narrowly on how participants are allocated to doses in real time as you move through the trial, and how at the end of the trial are 1 or more doses going to be selected for further study. A little bit of background, I'm part of the Center for Early Phase Trials at the University of Virginia. That's a group of us over the past 15 or so years have been dedicated to the design and implementation of novel early phase trials, and listed out the members of that trial at UVA and at VCU, mainly to acknowledge that most of what I'll talk about today is joint work and collaboration with my colleagues in the center. Over the past years, we've published a number of trials involving novel dose finding. We have many more ongoing. Certainly don't expect you to absorb what's on this extremely busy slide. Really wanted to put them in there for future reference in case you want to look back at some of these designs being put into play. All right, so if you do go through a lot of the trials that we've done, published or ongoing, you can see that the trials vary quite a bit. They vary in the type of agents. Some involve cytotoxic agents, some immunologic, some targeted agents. Those trials also vary quite a bit in their objectives. Some of the studies are toxicity only, where we're looking to identify a single combination or a set of combinations with acceptable toxicity. Some of them are safety and quote efficacy trials. And because I wanted this to be a very general presentation, I'll use efficacy in a very broad sense. It could mean a biomarker signal. It could mean drug activity. It could mean clinical, partial, or complete response. Any indication that you want to use to establish the efficacy of the combination. All right, so just a couple of examples. This is an example of adding a new agent to existing chemotherapeutic regime. This was funded about a year ago in an NIH grant where we were taking a monotherapy and moving it into combination trials seamlessly. And the intent of this was that the new agent would improve the efficacy of the existing chemotherapeutic agent without creating more toxicity, and in fact, maybe allowing you to use less of the chemotherapeutic agent to have the same level of efficacy. A second example, one that I'll talk about much more today, is this ABT-199 trial, and that was looking at combinations of venetoclax and ibrutinib, venetoclax at 2 levels, ibrutinib at 3 levels, and that was a study that had both safety and efficacy endpoints. And it was designed based on the assumption that the toxicity, the DLT probabilities would increase as you moved across rows, increasing the dose of ibrutinib, or up columns, increasing the dose of venetoclax. The efficacy for this study was objective response, partial or complete post-treatment, and it was designed with the possibility in mind that efficacy might plateau, might not occur at the highest doses of the two agents. So those two examples, and if you look through the list of examples on the previous slide, I think should convince you that every trial has unique features, not only in the type of agents and objectives, but dealing with issues like patient heterogeneity, early versus late toxicities, a mismatch in the timing of when toxicity is measured and when efficacy is measured. But the one thing all these trials do have in common is that we endeavored to tailor the design to the specific agents, the specific endpoints, and the specific objectives of the trial. Right? And so rather than focus on any single trial as a case study, because they are all so different, I'm going to kind of use a generic example to talk about implementation, how you can use safety and efficacy in allocations as you move through the trial, and what process is required to actually implement these designs. All right, so here's the general setup. Just two generic agents, A and B, could be cytotoxic, immunologic agents, whatever. And let's assume we've already decided on the hard decisions about that we're interested in 4 doses of A, 3 doses of B. We know how we want to define a DLT. We have a threshold for toxicity, and we have some measure of efficacy for those combinations. We're going to design this trial thinking that the agents in combination might have low toxicity, but that's something we're going to try and establish as we move through the trial. And we're going to at least allow the possibility that the efficacy might plateau. And so we're not going to assume that efficacy is going to increase monotonically with dose. All right, so how can we do this? How can we incorporate efficacy into this trial? Well, one way, and this was actually the way it was done for that monotherapy combination therapy study that I mentioned a few minutes ago, efficacy can be used at the end of the trial. That trial was designed that we would make our allocations based on toxicity only, and at the end of the trial, we would have presumably a set of combinations with acceptable toxicity, and from that set of acceptable toxicity combinations, at the end of the trial, we would evaluate efficacy and see which, if any, of those combinations we would want to move forward with. All right, so that's an example where we're going to allocate based on toxicity, and then at the end of the study, make decisions based on our measured efficacy. A second way of doing this, and what I'll spend more time talking about today, is incorporating efficacy as you move through the trial in, quote, real time. So safety and efficacy are going to be used in the allocations during the trial. And there's at least a couple of ways of doing this. Two ways of doing this are to create some kind of utility, to have some kind of weighting of different levels of safety and efficacy and assign that to the combinations, or what we have done more often is what I call safety first, and I'll describe that in just a couple of minutes. And within either of those ways, the safety first paradigm or this trade-off or utility paradigm for combining safety and efficacy, you can include elements of randomization, or you could always try and target what the current best dose looks like. Or have mix and match, where you'll do some element of randomization and some selection of what looks like the best dose. All right, so if you're going to use toxicity and efficacy in allocations, we'll assume that at any point in the trial for every combination, there will be some information. There'll be information on the number of participants that have been allocated to that combination, the number of participants that have experienced a DLT on that combination, and then a measure of efficacy for participants on that combination. And again, I'm going to use a very general notion of what efficacy is. So for example, in the safety first paradigm, what we're going to do is use the current data to estimate which combinations have acceptable toxicity. And this is a completely made up example, just to illustrate the ideas without the mathematical detail. Let's say we've been rolling through the trial and we've accrued 20 participants, and this table on my left, or your left too, I guess, is the number with the DLT and the number treated under each of the combinations. What we're going to do is use a mathematical model to estimate the DLT probabilities based on those observations. And the method of estimation, which I haven't really specified in any detail, suffice it to say for this example, the method of estimation guarantees that the estimates increase as you move up rows, increase the dose, or move across columns as you increase the dose of the second agent. And for purposes of the example, I'll say we've chosen pre-study an acceptable threshold of toxicity of 0.25. And so with these estimated DLT probabilities and with this threshold, we can separate the combinations into those that have estimated DLT probabilities less than the threshold, those in green, and those with estimated DLT probabilities greater than the threshold, those in red. And we often call this putting up a fence, kind of a dividing line between the currently estimated safe combinations and currently estimated unsafe combinations. So there are lots of ways of putting up that fence. There's lots of ways of deciding on that contour. Our group, in our experience, we've tended to use this partially ordered CRM method, partially ordered continual reassessment method. We use that in several studies. For larger grids of dose combinations, we found it convenient to use a method we call CDP based on this 2004 paper. But I'd be remiss in my duties as a speaker if I didn't point out that there are a lot of ways of figuring out where to draw this contour. There are model-based parametric methods. That is, you use a specific mathematical model to try and estimate the probability of a DLT at each combination. This is not an exhaustive list, these 4 papers. There are many others. There are model-based, quote, non-parametric methods where you're not using a specific mathematical model. You're just using information about the ordering. The ordering meaning if I increase the dose, I would expect to increase the probability of a DLT. Right? So it doesn't use a specific mathematical modeling, it just uses the ordering. And so I mentioned the CDP method. There's also this method by Mander and Sweeting called the pipe method. And basically what it does is lay out all possible contours and decide which contour seems the most likely given the current data. And you're all probably familiar with some interval-based methods. There are versions of interval-based methods like Boeing, which would help you draw that contour as well. All right, so however you decide on drawing this contour, wherever you decide to put that fence, this safety first paradigm says, we're going to allocate the next cohort of participants by choosing among the acceptable doses. We're going to choose among the green boxes. And you can do that in any number of ways. You could target which acceptable dose has the greatest estimated efficacy currently, or you can include some element of randomization. And the element of randomization could be done differently depending on your goals. Early on, we used to emphasize dose exploration. And so the randomization would randomize inversely proportional to the number of participants on a combination, kind of slanting the randomization towards acceptable combinations that have been used less. And so try and spread out the information. More recently, we focused primarily on having the goal of efficacy, and we would randomize in proportion to the estimated efficacy. So try and slant the randomization towards combinations that seem to have the greatest level of efficacy. So again, completely made up. An example, let's say we've already decided that the green boxes are the acceptable set, and we run some kind of mathematical model that would allow a plateau, and we come up with these estimates of the objective response rate in each of those combinations. An example of allocating with respect to efficacy would say, let's put next participant on A3B1 with probability 0.3 over the sum of those other probabilities, and so forth and so on. If you were going to target the combination with maximum efficacy, you would go to the combinations with estimated efficacy of 0.4, dose A2 and B2, and dose A2, B3, and you might randomly choose between those two. All right, so once you've completed that randomization, you're going to treat the next cohort at the selected combination and just keep repeating. You're going to get new information from that participant, which allow you to update the toxicity information and the acceptable set. You're going to get efficacy information from that participant or cohort of participants, which is going to allow you to update the efficacy and the randomization probabilities. And you just keep looping through this cycle until you hit a stopping rule. And the stopping rule could be as simple as a maximum number of patients, or sometimes we've used a rule that says we're going to stop when we hit a maximum number on any single combination. Either of those can be used. All right, so what does it take to actually implement one of these? We've heard the importance of communication among different groups, and I think that is absolutely crucial. It takes a coordinated effort between clinicians, statisticians, computing personnel, data management, beginning from the design, where the clinicians and statisticians would work together to tailor the design to the specific disease, the agents, and the endpoints. If you're going to do real-time adaptive allocations, you need good up-to-date data, so you need data systems that would allow that. You also need to be able to reproduce assignments. And so you need to be able to archive the data set so that if you go back at the end of the trial and say, why did participant 15 get A2, B2? You'll have the state of the data set at the time participant 15 was allocated, and you can reproduce that assignment. And a shout out to Gina Petroni for creating the data systems that would allow us to do this. Many of the things that we do are based on fitting models, and so you need software and computing to do that. And I think maybe most importantly, you need processes and communication. You need structure in place to make sure that the next dose allocation is available when the next accrual arrives. And so in terms of a schema, you can think about the timeline pre-study initiation and post-study initiation. Before the study, statisticians and clinicians would get together and develop the endpoints and objectives of the study, tailor a design, whether it's a new design or an existing design, to meet those objectives, evaluate the properties of the design, and incorporate that design into a protocol, including any additional rules as needed. So what's going to happen if two participants show up a day apart and you have to assign somebody before you have full information on the previous? These methods can handle that kind of situation, but it needs to be spelled out in the protocol. Once the study begins, the way it's worked for us is the clinical team would notify the statistical team about a potential accrual. That would trigger a pull from the database, a quality check to make sure that that data is up to date as possible, archive of that data set, and then the use of that data set to make an allocation based on the current data, which then gets fed back to the clinical team in order to do the allocation. And then this cycle would just continue throughout the trial. So I said earlier that we've tended to use model-based methods, the POCRM and sometimes CDP. That's been our preference because of the flexibility that they afford. It is possible that a participant won't receive the planned dose combination, either in rare cases because of some kind of error, but more likely because of a participant or clinician choice. Sometimes there's adjudications of DLTs. Sometimes what we thought was a DLT upon medical audit or medical review will be revised to a non-DLT or vice versa. Or sometimes we've had protocols where we wanted to incorporate toxicities that occurred outside the initial window for evaluating DLTs. We wanted to incorporate those into the evaluation. And so model-based designs handle these complexities without much trouble. They're just going to use the current state of the data to make the allocations, so forth and so on. So they won't have any trouble with this. All right, so in doing this, though, we've also found the importance of evaluating the properties of any novel design via operating characteristics. And what I mean is we would lay out plausible scenarios, so-called truth models, for the DLT probabilities and the efficacy, run the simulated trials many times, tabulating features of the design, the number of proportion of times it selects the best or an acceptable dose, which you'll know because you've set up the truth model, and other characteristics of the design, like the proportion of DLTs relative to the threshold, or the number of participants allocated to optimal or acceptable doses. So here's an example from the design paper that went along with ABT 199. There were many more scenarios evaluated, but you can see we're in the process of laying out various truth models and evaluating how does the design perform under different scenarios. So the operating characteristics do have the benefit of helping you decide among alternative methods of doing things. As I mentioned, there are lots of different ways of, say, putting up the fence, or different ways you can do the randomization. And these operating characteristics can help you decide between those alternative choices. Another value of creating operating characteristics is it forces specificity. You really can't program up ad hoc choices. And so the process of creating programs to do operating characteristics often forces you to recognize that you have to make decisions in certain situations, and forcing you to be specific about what those decisions will be. The other thing that we found very helpful in implementing these novel designs is observing individual trials. So not only the average behavior and operating characteristics, we would track how allocations move around combinations based on the current observed efficacy and toxicity data. And sometimes this helps identify issues with the design that may not be apparent in the average properties displayed in the operating characteristics. And this we found to be a very good tool for communicating what these designs are doing to our non-statistical colleagues. So you show them this is what the current toxicity data looks like. This is what the current efficacy data. This is what the design will tell us to do next. Does that make sense? And we have found that to be a very helpful way of communicating what these novel designs are doing. All right, so in summary, there are many designs for combinations that have been proposed, and they do have advantages over traditional MTD-only methods, but I certainly don't want to give the impression that everything's been answered. There are still a huge number of unanswered questions, and this is an active area of research. But I think that the work that's been done so far, leads me to think that the goals of Project Optimus, can be achieved with these designs. Features of these designs include the fact that the objectives will determine the design, not the other way around. They're tailored for specific agents and trial endpoints. It's important with any novel or even existing design to do a careful evaluation of the properties. And maybe the most important property is that these designs can be put into practice. They can be implemented. I'll stop. Thank you.
Thank Dr. Calloway for this insight presentation from statistic considerations. So our next presentation will be delivered by Dr. Dean Bodino from Takeda. So his presentation, he will discuss model-based approaches for dosage optimization of anti-cancer drug combinations.
Thank you very much. I'd like to thank the organizers for inviting me to present at this conference workshop. So let's just jump right in. So we're talking about oncology combinations, and this is an example of a pattern I think we tend to fall into when we're running trials real time. So you might have a single arm study that tells you how many patients in each dose cohort at the bottom, given by the green and red pie chart. So the red represents dose limiting toxicities, the green represents the proportion without. And so you might end up picking a dose of, say, 8 milligrams per day for drug A here as a maximum tolerated dose. You do similarly in a separate study, and you end up with, can't see it from here, but maybe 800 milligrams. And then when you start doing the combination, it gets tricky, especially when you have novel. There's a lot of degrees of freedom here, right? So you might drop down one dose below the MTD of drug B and then start titrating in more drug A, or you might do it the other way around, try to sneak up on it the other way, and you end up, next thing you know, you have 2 MTDs, and this is what our stakeholders get very nervous about. Which is the MTD? Well, it turns out there isn't one MTD, right? The mathematicians here will know that there's actually a whole constraint curve, as Professor Conaway just explained. There's a whole infinitely many, if you will, maximum tolerated dose combinations when you have a 2-drug combination. So what do? Well, one thing we propose is the general idea is along that toxicity constraint curve, so in that green tolerable region, you find the dose that maximizes the efficacy, exactly how Dr. Conaway just described. So looking at this from the side, like thinking of this as a surface, you have an efficacy surface, that's the green surface there, a toxicity constraint curve that restricts you to this tolerable region in red. And then you want to find the point along the fence, exactly the same terminology. I thought I made it up, but okay, that's fine. So along this constraint fence that maximizes your height on that surface, right? So the good news is, as long as you don't have really weird functions for your safety and efficacy, the optimum should occur along that fence, right? So the first time I ever worked with combination data, this was PD data, actually it wasn't safety and efficacy data this time, but I started using these 2D smoothing functions, and you don't want that because it doesn't have the kind of behavior you need. It doesn't have any pharmacologic plausibility at the end of the day, and you get these weird duck shapes that are hard to believe exist in nature, except for ducks, of course. Right. So 5 years ago, at a very similar workshop at the FDA MIDD workshop, I presented this example, and I'm just going to go through this case study in one slide because I want to go through newer stuff this year. So basically we had these two drug combination, both kinase inhibitors, TAC117 and TAC228. And the question is, which clinically tolerable dose pair gives us the biggest anti-tumor effect? So what we had was mouse data on anti-tumor effect across those little red and blue dots that you can see scattered around the XY plane there. And that let us characterize an efficacy surface. We didn't have reliable clinical efficacy data yet, although we had clinical safety data. And so the clinical safety data, if you look at the exposures, each patient is one dot there. The green dots are patients' exposure pairs for a patient that did not have a dose-limiting toxicity, and the red dots were those that did have a dose-limiting toxicity. So we fit this brown surface to the safety data, and you get this bright blue line that defines the 25% probability of toxicity. So you overlay that blue line, I'm not showing in this example, but what we found out was that for this particular example, the most efficacious combination was actually TAC117 alone at its MTD. And so that was the result of that particular study. So going on, and this is an example of some functions that we used in this particular example. You don't have to have the same function, right, for safety as for efficacy. So the growth rate inhibition function has some interaction term on the output, whereas the toxicity function has interaction terms on the input in dose or concentration space in this case. So today's presentation is more about a follow-up which involved newer methods that we updated. First starting with the methods that we had here is a slightly different scenario where we had two drugs, both small molecule inhibitors, very sparse phase 1 data in the combination, exploring various combinations. Also dose-limiting toxicity observed, and we only had historical summary level data on the monotherapies, so cohort by cohort, not patient by patient. So the synthetic surrogate that we're running today is drug X and drug Y, we'll call them. The trial design is this one. Has anyone ever heard of this trial design? Okay, let me explain it. I just coined it today. So MD gut refers to the gut instincts of the MD that's in charge of the trial. Now, has anyone ever experienced a trial like this? Okay, all right, a little better. All right, so we have sparse overall response rate data, we have sparse tox rate data, and then we just jump in. I thought there was one more animation, but that's okay. So this is the truth model that we never know in actual situation, right? So this is just the particular parameter set leads to a toxicity surface that gives you a slightly outward bowing toxicity isobol at, I think I'm using 30% as the target toxicity rate. And the true efficacy surface is in this blue-green color map. And you can see that it's quite synergistic in efficacy. But, so just keep in mind that the underlying true values that went into generating the synthetic data that I'm going to show you next is 40 milligrams of X and 500 milligrams of Y. So first step is you plot the raw data and you familiarize your stakeholders with the color maps that you're using. I'm sorry, this is my third, I think I've switched color maps 3 times already, but I'm going to stick with these color maps for the rest of the presentation. So you can see the number of patients is proportional to the size of each circle, and the color of each circle is proportional to the probability of toxicity. Similar with efficacy. So the next thing you do is you fit your model. And so, first of all, I just want to take an aside. There are many, many definitions of synergy. And I assert, based on an interesting paper that Dr. Chow presented a slide from this morning, that there could be more definitions of synergy than there are actual examples of synergy in oncology drug combinations. So, what it comes down to isn't whether or not you have synergy and efficacy in is whether there's essentially more synergy, regardless of how you define it. It's whether that efficacy surface, that toxicity curve, lets you access a superior efficacy when you move around on the efficacy surface. So I mentioned before, pick a pharmacologically reasonable interaction model. So I started this from Greco's paper, but we found that it was having this sort of, on the bottom left, you can see this sort of Pringles kind of shape going on, where it goes up and then starts coming back down as you get to really high doses. We don't necessarily want that in a pharmacologic model. And so what we did was, what this does have the nice feature of being a hill function in each of the monotherapy directions. Okay, so that's a popular pharmacologic function. What we ended up doing is we modified it. And in fact, I don't remember modifying this. So if anyone knows where this was published, if it was published somewhere, it seems a little too clever, but it works. It makes it monotonic. And so that's the function that we're using in this example. Okay, so we fit these functions. In this case, it happens to be the same function to both the toxicity and efficacy surfaces and to the toxicity and efficacy data. What you get is this bright pink curve. That's your maximum tolerated dose curve in dose space. And then we transpose it over to the efficacy surface on the right. And what we get, I'm just going to review some of these concepts with some animations. We're going to ask, right, what's the predicted efficacy as we move along this maximum tolerated, in this case, maximum tolerated dose fence. That's a typo. We're going to slice that fence along the edge and look at it from the origin. So that's what the graph on the right is. And so as we move back and forth on that fence, you can see where we are and try to find where that highest point is. And that gives you the doses of drug X and Y, giving you maximum efficacy and still tolerability. What can happen sometimes, and happened in that first example that I mentioned, is that you can have more synergistic toxicity than efficacy, and so you end up with no added benefit, so you're better off with one of the monotherapies. So you're not on the red curve, you're on the purple curve, and the best you can do on the purple curve is all drug X in this case. Okay, so going back to our synthetic data example, we end up with an optimum in this particular case that's very close to the edge. And in fact, there's no appreciable difference between where you would be if you had only drug X and if you had drug X and Y. That's just how it worked out in this particular synthetic realization of the data. And so that's the estimated optimal dose, which is notably a ways away from the input dose that went into generating the synthetic data. So to get a better sense of how uncertain we are, given, let's say, we don't know what the truth is, which is always the case, then we can simulate out, or we can bootstrap out where the optimum is going to be over different bootstrapping subsamplings of the original data set, the synthetic data set. And what you get is a couple hotspots, one in the middle and then one at the bottom right. And that's worth keeping in mind, that once you take a look at your uncertainty and you realize that you're not as certain as you thought you were by the point estimate. So now this is completely switching the question around. This is probability of achieving certain tox rates or better. So this is like a target product profile type question. So this is probability of achieving as a function of both drugs, your toxicity target, and probability of achieving your true response rate above some efficacy target of 55%. If we put these together, that's the important part, right? What's the probability of hitting both of those? Then you get this color map that puts you in a in the middle region in the dose space there. And so the dose combination maximizing probability of hitting the target product profile is in a different place than the point estimate for the optimal dose, which unfortunately is still in a different place than the true optimal dose. So then the question becomes how good was the design that we used to begin with, the sampling scheme. And so what I did was just and this should be done thousands of times, but I'm just showing two examples. So the first one was a data set we've been working with. The second is just another random sample from the same underlying truth model, but a different realization of it. And so that's what the toxicity data looks like. And now the toxicity curve is shifted a bit upward in data set 2. Same thing for data set 1 and data set 2. For efficacy, you get an updated efficacy isobologram, or sorry, the efficacy surface in blue-green that would put you in a different place. You finally, you remember what the optimum was. Is there a little purple star? Yeah, the pink stars show where the new optimum is on data set 2. It happens to be closer to the true optimum than we were in the last random draw of the data set. But we don't want this to be based on luck at the end of the day. So this is just comparing the probability of hitting the target profile, product profile in both cases. And the point that maximizes that is different in the two cases. And the second case happened to be even closer to the true optimum. But so the main thing though is it was a shortcoming of the design scheme of the sampling that made it highly variable in terms of what answer you would pick. So we need to do better designs like the ones Professor Conaway just shared with us, so that we get the optimal allocation of patients for not necessarily zooming in directly to the what we think is the right dose, but for informativeness as well. So combo MTD is not a unique number. So these are just some of the key points. Don't worry about synergy definitions. Get a function that makes sense for your plausible biology. This could even be a non-monotonic. If you have like a bispecific engager and you have a mechanistic reason to believe that it's not a monotonic function, then you would bring that in as well. Definitely use simulations to understand operating characteristics of your design, allocation, and analysis methods. Be clear what the key question is. So I showed you two different methods. They're not different methods. They're actually answering two different questions. One is, if you want to live life on the edge, the toxicity edge, what is the optimal, most efficacious dose along that edge? The other one is, if you want a reasonable chance of meeting both efficacy and safety bars, where should you dose in your pivotal study, for example? Certainly consider alternatives to MD gut sampling scheme, sorry. There are other methods that optimize information return for each point. And then finally, we did this, actually, as some of you might remember, we did this at ACOP a few years ago in the communication challenge. But before that, I went to the clinicians' oncology clinical research team meeting with brownies, and they're upset they couldn't eat them yet, but with brownies and fruit roll-ups, and had them make efficacy surfaces with Twizzlers for the toxicity constraint curve, and red and green sprinkles for the outcomes, the DLT outcomes for the patients. So with that, I'd like to thank my collaborators, first on the mouse efficacy and clinical safety data example, and then also on the fully clinical dose combination. And then at the end here, just a bunch of references and further reading from some of the papers I'd mentioned along the way. Thank you for your time.
Thank you, Dr. Bodino, for this excellent presentation. And thank you very much for your attention. And now we are going to have 30 minutes break. We will reconvene at 10:45.
Approach as well. So the purpose of the dose escalation is to select one or multiple dose regimens for further assessment in the next stage of the trial, which is dose expansion. And then dose expansion is to further assess the benefit risk of the candidate dose regimens, and then hopefully to select the optimal dose to be used for the next pivotal trial. And then the pivotal trial can confirm the efficacy and effectiveness of the optimal dose. So this sequential or stepwise process of dose escalation, expansion, and pivotal trial confirmation completes the full cycle of dose finding and optimization for a combination in clinical development. So I would like to further elaborate on the dose escalation step for the novel combination dose escalation, the novel dose finding. So in planning for the novel combination, there are two important decisions we need to make. So the first one, which dose to use for each of the agents used in the combination. And the second one, between the two combination agents, what is the order to increase the dose, or which agent should have the dose increment, and by how much? So in making those decisions, a number of factors need to be considered. So first, starting with the potential for PK drug-drug interaction. And if there's a potential for PK drug interaction between the combination agents, then the dose of the victim drug may need to be lowered for the combination. And then secondly, is there a potential to have overlapping or synergistic effect in the adverse event? Again, the dose may need to be lowered if there's an overlapping toxicity between the two agents. And then thirdly, the nature of the anticipated adverse events. So certainly the starting dose and also the dose increment need to be more conservative if there's involvement of irreversible AEs or AEs that are difficult to monitor or manage. And then an understanding of the individual contribution to both efficacy and safety by both agents is very important to help determine the order of the dose increment and also the range of doses to be explored for each of the agents as well. And then the steepness of the dose exposure response relationship can inform how much should the dose increment be. And then lastly, the anticipated difference in dose exposure response relationship between monotherapy and combination is also very important to inform the starting dose for the combination as well. In fact, I'm going to elaborate on this point in the next slide. So one may ask, so given all these questions, so where is the information to make those decisions, or where is the information come from? So I think, again, the prior information from non-clinical studies and also the monotherapy clinical studies should provide the source of information to address some of these questions. In many cases, in fact, we may not have the answers to all of these questions, but it helps to get as much information as possible, for sure. And then how would the anticipated difference from monotherapy in dose-response relationship may inform the combination dose escalation? So let's let's talk about two simplified scenarios. In the first scenario, the combination would only increase the maximal therapeutic response, but not change the threshold dose for toxicity. So in this case, the combination recommended phase 2 dose would be similar to the monotherapy recommended phase 2 dose. So accordingly, the combination may start at a dose closer to the single agent recommended phase 2 dose. In the second scenario, if the combination not only changes the maximal therapeutic response, but also sensitizes the dose for toxicity, in that case, the recommended phase 2 dose for the combination would be anticipated to be much lower than the monotherapy recommended phase 2 dose. So in that case, the combination may need to start at a dose much lower than the single agent recommended phase 2 dose. So in terms of the dose design for the novel combination dose escalation, so in general, there are two design options. The first one is what I would call fixed one as the backbone approach. So in this approach, we fix the dose for one of the combination agents to a certain dose and do the dose ranging only for the other agent. So in this illustrative example of combining two novel agents, A and B, so the dose of agent B was set to be the monotherapy recommended phase 2 dose. And then the dose ranging will be done for agent A, starting at 2 dose levels below the monotherapy recommended phase 2 dose. And the second approach is to do the escalation for both agents. So in this case, in this illustrative example, what I'm trying to show here is that, well, we start at 2 dose levels for both agents in the combination. And then in the next step, we may increase the dose for one of the agents. And then eventually, if we save, we may increase the dose to the recommended phase 2 dose for monotherapy for both agents. So how to select between these two approaches? So certainly, again, an understanding of the individual contribution of the agents to efficacy will be very important. But to give an example, the first approach may be taken if a backbone agent had a very narrow or very steep exposure-response relationship, and a reduce in dose would not be able to maintain the efficacy, even for the combination. So in that case, the first approach may be taken. And on the other hand, if there's a lack of understanding of the individual contribution to efficacy, then we may have to take the second approach, that we may need to do dose escalation for both. So that is the general considerations that I want to introduce for the novel combination. So next, I will present a case study from Pfizer in combining two investigational agents, drug X and drug Y. But these are two small molecule investigational agents currently under early clinical development at Pfizer for cancer indications. And each of these agents has completed the phase 1 monotherapy dose escalation in patients with solid tumors. And currently, the phase 1 combination study is ongoing. So in preclinical xenograft models, these two agents given together led to enhanced anti-tumor activity, as shown in this figure here. So this slide shows the phase 1 monotherapy dose escalation data for drug X. So the dose escalation included 4 dose levels, and the dose increments were 100%, 50%, 33%, respectively from the prior dose. And also the doses were given continuously twice a day orally. And the left figure shows the PK profile, the mean concentration time profile of the 4 doses. And we can see that there's a dose-dependent increase in exposure. And based on the safety, the safety told us that, well, at dose level 4, we started to see dose-limiting toxicities. So as a result, dose level 3 was selected as the monotherapy recommended phase 2 dose. And also from the PK profile, we can see that at dose level 2 and beyond, the exposure exceeded the predicted efficacious concentration through the entire dosing interval. And also we were able to predict the target coverage based on the in vitro pharmacodynamic data and the clinical PK data. So in the middle figure, we show that the target coverage for the 4 dose levels, again, higher dose are associated with a higher target coverage. And in terms of the AE profiles, neutropenia was observed to be one of the most common AEs observed for this compound. So based on the data from the dose escalation, we were able to develop a pkpd model to describe the neutrophil profiles. So the figure here shows the predicted neutrophil profiles for the 4 dose levels. Again, there was a dose-dependent increase in neutropenia based on this simulation. So this slide shows the phase 1 monotherapy dose escalation data for drug Y. Again, the dose escalation involved 4 dose levels. And the dose increments were 100%, 50%, and 33% again from the previous level. And the left figure shows the mean concentration time profiles of the 4 dose levels. So one thing we can observe from the PK profile is that, in fact, for the top 2 dose levels, we can see that there is almost a total overlap in exposure. So this is, in fact, very consistent with the predicted solubility limited absorption for this compound. So in that, the exposure going beyond the dose level 3 would not increase. And also based on the profile, you may see that dose level 2 had the overlapping profile as well. But what happened was that in one of the patients at the dose level 2, that patient had a much higher exposure than other patients. So as a whole, I moved the mean profile to a higher level. But there is a small difference between dose level 2 and dose level 3. And also in terms of the safety, so both dose level 3 and dose level 4 were very, very well tolerated. So in that case, dose level 3 was selected as the monotherapy recommended phase 2 dose. And also we can see from the PK profiles is that at all those levels, the exposure exceeded the preclinically predicted efficacious concentration. And then in terms of the target coverage, dose level 3 showed a little higher target coverage than the lower dose levels. And also for the AE, neutropenia was observed to be one of the most common AEs for this compound as well. And also we used the modeling approach to describe the neutropenia profiles for the different dose levels. And at dose level 3, in fact, the neutropenia was slightly higher than the lower dose levels again. And also one thing I need to mention for drug Y, that is this compound is predicted to be a time-dependent inhibitor of CYP3A4. And in fact, CYP3A4 is also involved in the metabolism of drug X. So based on the SynCyP modeling, we predicted that drug Y would increase the exposure of drug X by about 2-fold at all those dose levels, from dose level 1 to dose level 3. So obviously, when we think about the combination, combining these 2 agents together, we need to think about the overlapping toxicities. In addition to neutropenia, in fact, these both compounds showed some low-grade GI toxicities as well. So to determine a safe starting dose, so the first thing we did was trying to understand what would be the neutrophil profile if we combine the 2 agents together. Again, on the basis of modeling the individual agent data. So in trying to simulate what is going to happen to the neutrophil profiles, we made 2 assumptions. The first assumption is that these 2 agents will have an additive effect on neutrophils. And the second assumption, again, is that the drug Y will cause a 2-fold increase in exposure because of the potential drug-drug interaction. So based on those 2 assumptions, we simulated the neutrophil profiles for different combination dose level combination regimens for the 2 agents. And in the table below, we summarized the percentage of patients with any grade or grade 3 plus neutropenia for the different combinations. To just go into a little bit more detail. So you can see that, in fact, for drug X at dose level 1, whether it's regardless of the dose for compound Y, the grade 3 plus the neutropenia was around 20%. But when we increase the dose of compound X to dose level 2, then the grade 3 plus neutropenia will be around 40%, regardless of the dose of compound Y. So as a result, we decided to use dose level 1 for X as the starting dose. And also, as I mentioned earlier, so there's some concern about the overlapping GI toxicity as well. So in that regard, we decided to choose a dose level 2 for compound Y in the combination. So the final starting dose for this combination is X at the dose level 1 and Y at dose level 2. So in the dose escalation, the results from the first cohort showed that this cohort, in fact, was very well tolerated. There was no concern of DLT or any of the major AEs. And also, interestingly, based on the analysis of the data, we show that the PK profile from this combination for drug Y is very comparable to the monotherapy PK profile. So what was predicted as a twofold DDI, in fact, was not observed based on the actual data. So based on that finding, so we decided to increase the dose for both agents for the next cohort. So we increased the dose of X to dose level 2, and also increased the dose of dose level 2, of Y from dose level 2 to dose level 3. And then we observed that dose level, in fact, exceeded the maximum tolerated dose. So in 8 patients dosed, 3 observed DLTs. So what we did at the next step is trying to test the 2 regimens. So one, in fact, is further increase the dose of compound X, but reduce the dose of compound Y to the lowest level, dose level 1. And the second regimen we tested was where we reduce the dose for compound X to dose level 1, but we increase the dose of Y to dose level 3. So those are 2 regimens, one with a higher compound X dose and one with a higher compound Y dose. And then we tested both dose almost in parallel in the patients. The results showed that both those regimens were tolerated. And also based on the updated PK profile and also the neutrophil profile, we modeled the neutrophil counts for the different combinations used in the dose escalation. As summarized in this table here, we can see that for the last 2 dose regimens, in fact, the grade 3 plus neutropenia was 20% or less. So both were very well tolerated. So because at this time, we don't have a very clear understanding of the individual contribution of compound X and also compound Y to the overall efficacy. So we decided that we may move both regimen into the dose expansion. So the dose expansion, our plan is to enroll 30 patients per regimen in a head-to-head comparison. And then hopefully, based on the data, we're going to select the optimal dose. And this expansion is ongoing. And in terms of the criteria to select the optimal dose, certainly we're going to consider a number of factors. The first, of course, is the benefit risk for the 2 regimens, including assessment of the efficacy endpoint and also the safety tolerability profile. But also, we need to consider other factors as well in selecting the optimal dose. So the other factors can include, for example, the recommended phase 3 dose for each of these monotherapy in further development. And also, the targeted population for the pivotal trial as well will be considered. So the key takeaways from this presentation. So first of all, the novel combination dose finding needs to be informed by both preclinical and the monotherapy clinical data. And also it may involve exploring different dose regimens for each of the agents used in the combination. And then secondly, the combination dose escalation needs to consider a number of factors that I mentioned. So including the potential for PK drug-drug interaction, the individual agent AE profile, the dose exposure response relationships, and also the individual contribution to both efficacy and the AEs, to name a few. And then the optimum dose can be informed by comparative assessment of the benefit risk of 2 or more dose regimens. And also it should be done in an adequate number of patients as well. And then finally, so the case study also showed that the modern simulation tool is extremely important to enhance the efficiency and the confidence in dose finding. I would like to acknowledge a number of my colleagues at Pfizer for their contributions to this presentation. Jin Yang, Jerry Lee, Li Zhou, and Justin Hoffman, so they contributed to the case study development. And also I want to thank the members of the Drug X and Drug Y program teams for conducting the study and for getting the data. And also I want to thank all the trial participants for contributing to the data included in this presentation. Thank you very much.
Thank you, Dr. Yin, for sharing this very informative actual case from Pfizer. So our next presentation will be delivered by Dr. Simon from Sanofi, and she will discuss leveraging pkpd analysis to optimize the dosage of Isatuximab in combination with pomalidomide and dexamethasone.
So I would like to thank you, the FDA, for inviting me to present the case on Isatuximab. So multiple myeloma is the second most common hematological malignancy and remains incurable in elderly patients. This disease is characterized by the production of cancerous plasma cells in bone marrow. In most of the patients, this plasma cell produce a large amount of non-functional immunoglobulin called M protein. Isatuximab is an IgG monoclonal antibody that targets CD38. CD38 is a receptor expressed widely in multiple myeloma, while its expression is very low in normal cells. Isatuximab killed multiple myeloma cells via different mechanisms of action, including ADCC, CDC, direct apoptosis, and inhibition of CD38 enzyme activity. To support the drug development of Isatuximab, several modeling and simulation were performed from preclinical to market approval. I will start with the pkpd analysis in tumor-bearing mice. This analysis allowed to define a threshold concentration for tumor eradication in mice, which was used to guide the dose escalation in the monotherapy phase 1 study. So when the data of the monotherapy phase 1 study became available, we performed a population PK analysis, exposure response analysis, and disease modeling of serum and protein to support the dose selection of the monotherapy phase 2 dose. As a result, we have to modify the dosing schedule with the implementation of a loading phase instead of a flat every 2 weeks dose that was originally planned. So leveraging the information from the modeling of the monotherapy, we have implemented this new dosing regimen in the combination phase 1 study. We applied the same methodology regarding modeling and simulation to support the phase 1 dosing study in combination with pomalidomide. So leveraging this information, we were able to support the dose of this new combination. Then the dose was implemented in the phase 3 studies, which is ICORIA. It's the first combination that were tested in multiple relapse refractory multiple myeloma. When the data of the phase 3 became available, we performed a population PK analysis and an exposure response that were able to confirm the dose and then to support ICARIA filing. So now I will start with the pkpd analysis in tumor-bearing mice. So this analysis used the Isatuximab PK and tumor-bearing mice volume from 2 preclinical studies First, we used a single dose study in normal mice, and then a repeated dose study in tumor-bearing mice. A Simeone pkpd model for tumor growth inhibition was used and fit very well the tumor volume, as you can see on the graph, for the control and for the treatment, as you can see on an individual fit. From this model, we were able to derive secondary parameters, and one of interest was the target threshold concentration for the tumor eradication in mice, which was estimated at 129 micrograms per ml. And I will show you how this concentration was used in the first in-human monotherapy study. So the design of the first in-human study started with a very low dose because the Mabel approach was used. The dose escalation was divided into two phases: an accelerated escalation phase with one patient enrolled per cohort, and then a basic escalation phase where 3 to 5 or to 6 patients enrolled per cohort. The safety was good, and the MTD was not reached up to 20 milligrams per kg every 2 weeks. So 10 milligrams per kg every 2 weeks was chosen for the expansion phase because we haven't seen any dose response between 10 milligrams per kg weekly every 2 weeks and 20 milligrams every 2 weeks. However, we have seen an unexpected lower response rate in the expansion cohort. So to better understand the relationship between the dose, the exposure, and the efficacy, we have put in place the following strategy. So at first, we developed a population PK model with the phase 1 data. The simulated concentration were compared to the threshold concentration for tumor eradication in mice. Then we have performed exposure response analysis to characterize the relationship between exposure and overall response rate. And in parallel, we developed a disease model of tumor burden, which is the serum and protein. The serum and protein data were well predicted by a tumor growth inhibition model, wherein the serum and protein is controlled by two processes: a proliferation rate and an elimination rate. And we have seen that the Isatuximab exposure stimulates the elimination rate. So at first, we started with a preliminary population PK model and a 2-compartment with a parallel nonlinear and linear elimination from the central compartment was used to fit the phase 1 data. Due to TMDD in some patients, we have seen a lower exposure in the expansion cohort, which led to this lower expected overall response rate. So using this model, we have simulated the 3-dose regimen that were planned to be tested in the phase 2 protocol, as you can see on the slide. And the simulated concentration were below the concentration for tumor eradication in mice, as you can see on the 3 graph. The conclusion came: so do we have the right dose, or do we need a more intense schedule to optimize the dose? To answer to this question, we have evaluated alternative schedule by simulating Isatuximab concentration, overall response rate, and the change in serum and protein from baseline at week 10, 12, So based on the simulation, we have seen that 4 weekly administration together with a high dose of 10 or 20 milligram allowed to reach the concentration that we are targeting to eradicate the tumor in mice and to overcome TMDD. Higher response rate and greater decrease in the serum M protein were observed with this weekly administration at a dose of 20 milligram per kg. Therefore, the phase 2 protocol was amended and a fourth arm was added in the dose finding, testing 20 milligram per kg weekly at the first cycle, followed by every 2 weeks. When the data of the dose finding were available, we refined the model with a larger data set. So I will discuss first this exposure response analysis. We tested early endpoint because the time to first response occurred at 1 month. And CTRF at 4 weeks was found to be the best predictor of the response in the IMAX model. In addition to CTRF at 4 weeks, the percentage of plasma cell in bone marrow was added in the model. It is not surprising because a high percentage of plasma cell in bone marrow is synonym of a very active disease. So based on this final model, the value of the CTRF at 4 weeks to reach 90% of the maximal effect was 129.5 microgram per ml, which was very close to the concentration of the tumor eradication in mice. So we have validated our preclinical target. So based on the trial simulation, the highest overall response rate was observed with 20 milligram per kg given weekly, and the probability of success to reach at least 30% of the response was also found the highest with this schedule. To complement the population, to complement this exposure response analysis, we have performed and refined the tumor growth inhibition model with serum and protein. We confirm the effect on Isatuximab as a simulating effect on the elimination of the serum and protein. And based on this model, we saw that simulation were performed for different schedule of administration and confirm that 20 milligram weekly for 4 weeks followed by every 2 weeks gives the biggest decrease in serum and protein at week 8, 10, or 12. This model allows us as well to look at the loading phase, at the length of the loading phase, and the model demonstrates that there was no clear benefit of increasing the loading period beyond 4 weekly administration. So based on this analysis, we have implemented this new regimen with a loading phase in the phase 1 combination. So the phase 1 combination study with pomalidomide and dexamethasone used a 3+3 escalation phase. 3 dose levels were tested at 5, 10, and 20 milligrams per kg. 6 to 9 patients were included per cohort because 1 DLT per cohort was observed. So based on this dose escalation, the dose of 10 milligram per kg weekly for 4-week administration every 2 weeks was selected because we haven't seen any dose response for efficacy. So the same modeling and simulation strategy was applied to support the dose selection of the combination of Isatuximab with pomalidomide. First, I will discuss the exposure response analysis. The model showed that CTRF at 4 weeks was the best predictor of the response. In the final model, beta-2 microglobulin was added. Beta-2 globulin is also a tumor marker in several M diseases. It can be noted that we have seen an imbalance with beta-2 microglobulin in the different cohorts, and therefore it explains why we have no apparent dose response in the expansion phase. Based on trial simulation, the highest probability of success to reach the highest ORR was observed with 20 milligram per kg versus 10 milligram per kg. However, we have an overlap between the confidence interval. So when we look at the probability of success, to target a 60% response rate or higher, it was already very high for 10 milligram per kg weekly, and therefore it was selected over 20 milligram for the combination. So 10 milligram per kg weekly for 4 administration, followed by weekly every 2 weeks administration, was selected for the pivotal phase 3 study. Then I will move to the disease modeling with serum and protein. So the model included monotherapy and combination data. The serum and protein was modeled with a tumor growth inhibition, and pomalidomide and Isatuximab were found to stimulate the elimination of serum protein. As you can see on the graph, the simulation shows that there was no substantial benefit of decrease the serum and protein when you double the dose from 10 to 20 milligrams, which was not the case for the monotherapy, where we have seen a decrease of twofold when, at least a twofold when you increase the dose from 10 to 20 milligrams. So based on this result, we also confirm that 20 milligrams is not needed for the combination, and the dose of 10 milligrams per kg was selected for Icaria. And then now I will go to the confirmation of the 10 milligrams per kg using the Icaria data. First, the pivotal phase 3 was positive for the primary endpoint PFS. At first, a population PK model was developed with monotherapy and the combination with pomalidomide, and a time-dependent linear clearance was identified for Isatuximab with a linear clearance decrease when the disease status improved. So it was a justification to use early endpoint in the exposure response analysis. So CTRF at 4 weeks was found to be the best predictor of efficacy for PFS and overall response rate. Besides CTRF at 4 weeks, additional covariates were added in the model. So we had albumin and beta-2 microglobulin for PFS, and we added RSS, so there is a staging for multiple myeloma in the for the overall response model. So the main take-home message is the fact that we have seen we have a dose response, and the patient in the lowest exposure quartile are still benefited to Isatuximab treatment compared to the active control group, pomalidomide dexamethasone. However, the patient in the lowest quartiles were identified as patients with dose omission, leading to lower exposure. But these patients have also more aggressive myeloma characteristic, which makes them at risk more to develop AE, leading to dose omission, dose reduction. So these patients in the lower quartiles of exposure were confounding by their baseline characteristic and by the incidence of dose omission modification. So, and I will conclude that the modeling and simulation had a major impact on dose optimization and selection for the combination with pomalidomide. The modeling and simulation increased the robustness of decision-making during the clinical development. 2 approvals have been granted for Isatuximab in combination. One with pomalidomide dexamethasone using ECURIA data, and the second one with carfilzomib dexamethasone with IKEMA data. These two combinations were used relapse refractory clinical setting. And I will finish by thanking the patient and the investigator who contribute to this study. And I will also thank all the scientists from Sanofi who work with me on this journey. Thank you.
Thank you, Dr. Symbiont, for sharing the model-supported dose optimization for Isatuximab add-on therapy. So our last case study will be presented by Dr. Organization from Aztecs. So he will explore the clinical development and the dosage optimization for all decitabine and cytarabine combinations.
I wanted to thank the FDA and the organizers for the invitation, and delighted with the opportunity to share the experience for developing oral decitabine with cedazuridine. That's a brief outline of my presentation. I'll touch on our non-clinical setup going into the clinical stage for development and walk you through the phase 1, 2, 3 experience. Decitabine is approved for myelodysplastic syndromes. It's a heterogeneous group of clonal bone marrow stem cell disorders characterized by accumulation of mutations over time. And on the spectrum from left to right, the increase in disease severity goes up, as does the mutation burden, as can be seen, quite significant, seen with a very little frequency in AML, reaching for 50% or even higher. DNA methylation is abnormal in most cancers, and this is particularly relevant for epigenetic readers and writers that are often mutated in different cancers, and a number of genes are listed here that are relevant to hematological indications. The hypothesis behind the use of hypomethylating agents is with the treatment of this using this epigenetic reprogramming, you may see reversal of these aberrant changes and hopefully an effect on the disease. And DNA methyltransferase inhibitors, or hypomethylating agents as they are known, they have shown efficacy in hematological indications, MDS and AML. There have been anecdotal evidence in experimental setting in solid tumor indications, but the approvals are currently in hematological space. And these are the 2 agents in the class, azacitidine and decitabine. they both work by the mechanism of action in a similar fashion. Azacitidine, as you can see with a higher dose, does convert partially into decitabine before decitabine is phosphorylated further prior to entering the cell and being incorporated into DNA during the S phase, and at which step it irreversibly inhibits DNA methyltransferase. And this 10 to 20%, 10 to 20% conversion rate actually checks out if you compare the total cycle dose between azacitidine and decitabine. So azacitidine can be assumed to operate as a prodrug of decitabine. Both of these agents were initially developed and approved as parenteral drugs. However, of course, oral therapy is much desired because of the burden, potentially reducing the burden on caregivers as well as medical system in general. And unfortunately, the early experience of developing these as oral agents, as single agent, specifically with the case of decitabine, was not successful because of high first-pass metabolism mediated by cytidine deaminase that deactivates decitabine and further degradation then renally excreted. So our hypothesis was if we were to combine decitabine at a safe dose of inhibitor with cytidine deaminase, then that could enable or enhance the oral bioavailability of decitabine and allow us to develop orally at a reasonable dose. In essence, this was a proposal for a beneficially managed drug-drug interaction. There are a number of precedents for this when a PK enhancer, enzyme inhibitor, is combined with the active agent orally to enable higher bioavailability. And some examples are listed here. Now about cedazuridine, the enzyme inhibitor in this case. Research with inhibition of CDA goes back about 25, 30 years at least with the use of tetrahydrouridine, an old compound that also inhibits cytidine deaminase. However, tetrahydrouridine, THU, has a limitation in terms of its pharmaceutical profile. It's not very stable and requires much higher doses. Cedazuridine, with the code name E7727, was designed specifically to be more stable and have a potent inhibition of cytidine deaminase, and it's very safe and tolerated in toxicology evaluation in both species at very high dose levels, and well tolerated in subchronic studies of at least 3 cycle duration. And in our preclinical proof of concept studies in monkeys, a very low dose compared to those toxicologically meaningful dose levels of 1 milligrams per kilogram resulted in a 14-fold effect of increase in decitabine exposures. And this is the data we had, everything all on one slide going into the clinic. The experience with single agent decitabine at low dose levels that are clinically meaningful compared to IV resulted in very low exposures. And in monkeys here, a similar dose given alone, again, doesn't quite reach where we need to be. This is a bracketed range of AUC that we needed to target for our program. And the range was variable because it wasn't clear what number we should target, because the lower number was from the decitabine IV label, but the published data suggested exposures could be somewhat higher. And as you can see, at very low dose levels of cedazuridine, we very quickly were able in monkeys to reach that targeted clinical range for the AUC. It was also reassuring that continuous increasing of doses resulted in a relatively linear increase in decitabine AUC, suggesting that we're not achieving saturation at the dose levels we're going to be testing in the clinic. So our phase 1 dose exploration study had a primary objective to emulate pharmacokinetics of IV decitabine. And of course, secondary objectives are relevant as well for safety and clinical efficacy, as well as pharmacodynamics. As you would expect with a drug interaction proposal, it was very heavy PK study in phase 1. We dosed on day -3 oral decitabine alone, The thick arrows here show the days of serial PK collections when we had a pre-dose collections on the other days as well. On day 1, every patient received 1 day of IV decitabine to serve as the reference control for each patient. Starting on day 2, oral combination was given when we evaluated PK on the first day and the last day of the cycle. And Jean asked the questions earlier, can you get a pass for agents that don't have activity or are not expected to have activity, so you don't have to test them as a single agent? So we did propose not to do single agent cedazuridine studies. FDA agreed with us, but we did commit, of course, to dose on day 22 cedazuridine alone, just to show that interaction is only one way from cedazuridine on decitabine and not the reverse. And this is a progress during dose escalation. Oh, sorry. So decitabine, we started testing at 20 milligram. It was tested at 3 different dose levels. As you can see, without cedazuridine, very low levels. And we've set here fixed the IV exposures at 100%. The first dose level is cedazuridine. Second dose level mentioned is decitabine. The first 3 cohorts, we escalated only the inhibitor, and we reached approximately half of the desired exposure on the last day of the cycle. And then we doubled the dose of decitabine. That slightly overachieved. We dropped down the escalated to 30 milligrams decitabine. At the steady state on day 5, exposures approached the IV, but on day 1, they were lower. And these are the PK curves from dose escalation. The blue represents the IV reference. As you can see, the cohort 4, again, apologies, cohort 4 overachieved, and cohort 5 underachieved slightly. Here at 128% based on preliminary total cycle AUC calculation and 81% at the 30 milligrams. So this is what we actually were expecting based on our modeling predictions, and this data was supportive of taking the intermediate dose between the two, 35 milligrams, into phase 2. So we didn't test the 35 milligrams dose in phase 1. This is the pharmacodynamics of decitabine that manifests over the treatment cycle, which is defined 4 weeks, 28 days. Just a reminder, the dosing is only for the first 5 days, but the peak effect in maximum demethylation is not reached until about, in this graph, day 8, but in reality about day 10 or so, we're limited with sampling to only once per week. And as you can see, the effect is reversible, and for most patients are ready to resume treatment in cycle 2 on day 29. And these are the PK curves in phase 2. So in, compared to phase 1, we were able to confirm that based on our modeling predictions as well, that a steady state is reached with the second dose. So on day 2 and day 5 overlapped largely. Cmax for oral, as expected, was lower than for IV, and day 1 levels were lower. So this confirmed our proposed endpoint for phase 3, by the methodology of calculation that the total cycle AUC would take the 4 days of the steady state exposures, adding the 1 day lower exposures from day 1 and compared to IV. And of course, for IV, exposures are reproducible from day to day. This is a PK curve of cedazuridine, very low variability. Again, a very small accumulation from day 1 to the second day, and day 2 and day 5 are largely overlapping. And the steady state achievement is explained by looking at the trough levels. Of course, on day 1 with the first dose of inhibitor, the values are at 0, and consistent trough levels then are observed from the rest of the days of the cycle. That explains the consistent exposures from day 2 to day 5. And with this data, we approached the FDA for discussions at the end of phase 2 in terms of our design for phase 3 and registration strategy, and invoking the guidance from 1998 that has a scenario of considering development of a new route of administration for a drug for which clinical evidence, sufficient clinical evidence is available. And with this, our phase 3 design was a crossover design, and with the primary endpoint of total 5-day AUC calculation. And in the first cycles, patients received either a full cycle of IV decitabine or oral combination. And again, if the phase 3 design repeated exactly what we did in phase 2. And the second cycle, they crossed over to the other treatment. And from third cycle 3 and on, everyone received only oral drug. And secondary endpoints, of course, included clinical safety and efficacy, as well as pharmacodynamics. So very typical baseline characteristics for MDS population, newly diagnosed, median age of 71, 2-to-1 distribution, males, females. And importantly, for body weight, we had a wide range of body weights. The difference here, of course, IV decitabine is dosed by body surface area, as opposed to oral combination when we proposed a fixed dose regardless of body size. So this was important to us to capture a representative spectrum of body weights in the population so we could analyze properly and validate the dose selected to be appropriate for the population. And this is the top line data from a primary endpoint of the trial. It was met with high confidence, 98.9% oral versus IV, with very tight confidence interval and relatively low interpatient variability, considering that we're comparing two different routes of administration here. And you could say based on this that the study was highly overpowered, and that's accurate. We could easily meet the PK primary endpoint of the trial, which with a much smaller number of subjects. However, we really needed the numbers because cedazuridine being a new chemical entity, we needed to generate sufficient number of safety data set for submission. This graph is meant to help you appreciate the variability of oral versus IV. We're very pleased that the variability for oral decitabine, when it's dosed at a fixed dose regardless of body size, was only slightly higher than IV. And the CDA status, which varies from patient to patient, is, we believe, the main or one of the chief factors contributing to variability for IV. So by inhibiting cytidine deaminase, kind of normalizes across the population and reduces overall variability, while the rest of the factors such as body weight and body size then contribute to the variability for the oral drug. As you can see here, again, that's a steady state effect. Phase 1 values are lower in general, but phase 2, the day 1 and the day 2 and day 5 largely overlap. The combination of all these data points allowed us to calculate a total cycle AUC. And a pharmacodynamic assessment over 2 cycles confirmed that AUC is sufficient to replicate what we see with IV. And overall survival, of course, so with to be interpreted with caution. We didn't have a control arm in this study, single arm study, but data could certainly be compared to what's historically available for decitabine in this population, and data look quite favorable with almost 32 months of survival and overall response rate up to 62%, of which 22% were in complete remission. On a very busy slide on safety, but the important message here is there were no new safety signals identified that could be blamed on cedazuridine. As I mentioned earlier, we had a very high safety margin for cedazuridine. All the observed adverse events were very consistent with what one would expect with IV decitabine. So this is the model that we presented different iterations of this at this meeting over the years as we honed in to have a final version of it that performed very well. We used it recently for doing simulations to support the development in Asian demographics, as you would expect with a lower body size that would be expected to receive a lower IV dose. And as a consequence of that, somewhat lower oral dose would be needed to replicate what we achieved in the North American population. And recently we used a model for pediatric simulations as well. And again, for the most important covariates, sex, body weight, as well as a mild, moderate hepatic impairment did not have any clinically meaningful effect on a PK of decitabine or cedazuridine. The mild or moderate renal impairment, and we had such subjects in our phase 2, phase 3, had an effect, as you would expect, on cedazuridine, and the cedazuridine was also the most influential covariate, as you would expect in a drug-drug interaction combination, the most important factor on exposure of decitabine. And we evaluated the body weight effect of body weight on exposures, very different angles here, a simple correlation between IV and oral to show the highest and lowest 10% in the population, and higher body weight patients in green here would be expected to have lower exposures on average. However, this effect is in the population is quite muted. The trend is definitely there, but there is no obvious case of outliers overrepresented in one or the other groups. And the exposure response exposure safety analysis looked at this very carefully, and we didn't see anything that would suggest the treatment for lower body size patients should be at a different dose, so everyone receives the same dose. There's some post-marketing trials ongoing in a renal and hepatic impairment, specifically in the severely impaired renal patients and moderately and severe impaired hepatic patients. And another food effect study post-marketing was completed recently to show meaningful reduction of decitabine exposures with dose. So this combination is dosed at fasted state. Relevant to dose optimization, because there's only one dose approved at this time, dose reductions are implemented by the way of reducing the number of days in the cycle from 5 days to 4 days and further to 3 days. And there's another reduction option available, taking advantage of this steady state effect, day 2 versus day 1, by giving the 3 doses on, for example, Monday, Wednesday, Friday, as opposed to Monday, Tuesday, Wednesday. That achieves another level of reduction. And in our phase 1, phase 2, we had the number of patients continued to 4 or 5 years on the oral therapy, and all of them were on reduced dose, and thus a reduction is expected in most patients with the treatment of HMAs because their bone marrow status at the start of treatment is different compared to when they achieve base response after continuous treatment. Just to summarize, this approach was successful to develop oral decitabine based on pharmacokinetic AUC equivalence against IV decitabine in phase 3. And we submitted as part of Project Orbis to US, Canada, and Australia. And the approvals were received in the middle of a lockdown. Actually, it was very nice for the patients to get the option to get the treatment at home without traveling to the clinics. And more recently, we completed exactly the same design phase 3 in a different population because of the approved label is different in Europe. And recently approvals in EU and UK were also received, and additional submissions globally are ongoing. Clinical and safety efficacy data were similar compared to historical data available for IV decitabine, and we have an ongoing pediatric investigation program. It's a very busy slide. I'll only highlight at the bottom here from a first patient dose to registration. The first approval was approximately 6 years. And we had a little bit of slow start. This could have been done a little bit faster even. Of course, we're all very happy about that, and an acknowledgement. So big thanks to the patients and their families and caregivers with these rigorous PK assessments. And I felt very guilty designing this because understanding what they have to go through. But again, the reward for the patient was receiving oral therapy from cycle 3 and on without the need to go back to the clinics for the infusions. And the principal investigators and the sites and personnel and very long project team for the program, as well as a Metrum group with whom we worked on the model for this program, and some of them are in the audience. Thank you for that as well. Thank you.
Thank you, Dr. Ognisha. So for this informative case of model supported dose selection for combination with PK booster. Now we close our presentation. I will pass the podium to Vijay for the panel discussion.
Thank you everyone for the presentation. So in addition to the speakers, we also have Dr. Jiang and Dr. Verheyo from the FDA will join the panel discussion. So just a quick announcement. This side mic on this side of this room is not connected to the broadcast. So if you have questions, please use the two mics.
It has been corrected. Now we are up.
Okay, thank you. All right, so please feel free to come by and get to the mic for questions. But I'll probably start the first question perhaps to Dr. Conway, who gave a very good initial presentation on design principles. There is a nice balance between how drug companies want to run their trials and get the results very quickly, and now you're proposing look at the results and reallocate designs. So what is the cost of that? And I'm assuming it's a cost at both at the patient level and at the design level of the, or actually for the company who's conducting the trial.
Yeah, that's a very good question. I mean, for these studies, if you're going to do adaptive allocation, you need both short-term measures of toxicity and efficacy, and you need those short-term measures to also predict the longer-term, more definitive endpoints. That we were fortunate in the ABT 199 study is we used 1 month. It was actually a treatment course of 6 one-month cycles. We used the first cycle to determine DLT and initial clinical complete or partial response, and then use that information until the 6-month data came in. And so we were using 1 month data until it got matured. And then when the 6-month data was available, that got substituted into the database for the 1 month. And so we were always using short-term information supplemented by longer-term information. And so in that case, the adaptive design, given the accrual rate on that trial, didn't really slow us down. But I think in other cases, yes, you absolutely need short-term information, and you need that information to be a good predictor of the longer, more definitive endpoints.
Thank you. And just a quick follow-up on that. You mentioned in your presentation the use of data systems and continuous integration. So is there some sort of a technology that you're using to allow folks to run these more seamlessly, or is this something that we all have to consider?
It wasn't anything. It was mostly homegrown. It was mostly what we did to make sure we had the information available so that we could turn it around to make recommendations for doses to the clinical team. So it was much, I would say, hands-on work. Literally, when I said pulling the data from the database, that's what would happen. Every Friday, there would be a data pull. Whether we had accrual, whether we were anticipating an accrual or not, we would do it more often if we were anticipating an accrual. So it was very manual, very hands-on, and it would be nice to streamline that, but that's not what we did.
Yeah, thank you. Right.
Yep. Hi, Mohammed Tariq, Pumas AI. Thanks for the nice presentations. Sorry, I have a bit of a cold, so my voice might be a bit off, but. So I have a background in computer science. So I'm interested in kind of the algorithmic open questions, let's say. So do you see a lot of areas where there could be new algorithms or new methods, maybe from machine learning or elsewhere, that could improve those optimization and even the model development phase of those optimization?
Okay, yeah, I'll take a shot at that. I think there's always room for improvement. I think I have my own workflows that may or may not be are probably not optimal, right? And so, in terms of, I think the optimal information content of the next set of patients to be allocated is, I think, still an open problem, at least in my mind. So. I actually like Dr. Conaway's allocation based on likelihood of response, but that informativeness and likelihood of response don't necessarily track with each other. And so there might be a way to spread out those doses in a way that lets you have more confidence in your final dose combination.
Anybody else?
I would just like to echo that there's always room for improvement. There's always room for improvement. We tend to use fairly simple models. It was pointed out this morning, we're in a fairly data sparse environment, and it's often difficult to fit complicated models, but no, there's always room for improvement.
All right, just a quick follow. Yeah, sorry.
Go ahead.
Thank you for the question. So the modeling simulation is always an emerging area where we'll have the traditional PK analysis. We know like nowadays we incorporate more like magnetic modeling, pkpd, QSP modeling, and we have so much information. It probably already have been done with some other drug, but shares similar information, and this kind of machine learning, big data may help us to fit in the model to develop a modeling platform to help us make an informed decision, even with very minimal information from the current drug, but probably we already have a lot of information that can be shared and informed.
Just a follow-up maybe comment. So yeah, I think it's maybe the field of semi-mechanistic models or scientific machine learning, as it's rebranded, I think these days, could be useful where you can use both mechanistic and empirical components in your model. But just another follow-up question is, do you think there's room for innovation or I guess changing the structure of the decision itself instead of just one dose, maybe sort of like a decision tree of if you see this, pick this dose, if you see that, pick that dose, like a more sophisticated policy for dose selection. It's like poor man's precision medicine, I guess.
Thank you. That's a wonderful question, and we always try to find the best dose for the right dose for the right patient at the right moment. So we are collecting the data, and definitely we want to make an informed decision to find the best approach for the patient.
Thank you. Raj.
Raj Madabushi from Office of Clinical Pharmacology, FDA. Very insightful presentations and practical examples. So I had a quick question with respect to these early dose escalation and checking the combinations, either from Dr. Conway, your presentation, or from the examples that were presented, I think specifically, Dr. in one of your examples. The way I understood was decisions are made on point estimates, and this is a data sparse environment we are talking about. And given that these are early in development, how are we accounting for uncertainty in those point estimates as we are moving forward, and what kind of quantitative approaches that we are invoking to be able to reduce uncertainty and bring more confidence? Any insights into that?
I think it was
okay. Yeah, I mean, in the presentation I gave, there was an emphasis on using bootstrapping to get a sense of our uncertainty. It doesn't reduce uncertainty; it lets us know our uncertainty, which I think is better than going in blind, right? So, yeah, so that's one class of techniques that I think you could apply generally to any optimization algorithm you choose. You just bootstrap on the samples that you have, and that's always a great way to mitigate the risk of believing too much in your point estimate.
And I would agree with that. I think you always need to be cognizant of the tremendous variability in those point estimates. But at some point you do have to start making decisions, and so you could make decisions based on adding variability to that, the upper limit of a confidence interval or the lower limit, depending on what you're looking at. So you could certainly change the rules to try and account for that variability. But at the end of the day, you're still going to have to make a decision, understanding that variability is going to be part of that decision, no matter what you do.
Dan.
Yeah, I can also add to that. I think there certainly is a very common challenge in early stage in that the sample size is limited, and there can be the patient population can be very variable, and also all the heterogeneity in the population as well. So certainly it's a very common challenge in early stage. But I think certainly there's some other tools that we can use to address this challenge. So one of the tools can be modeling simulation, for example. So we try to use the modeling to integrate all the data. So sometimes you may have a point estimate for a certain dose level, but when you integrate all the information from the different dose levels and also integrating the time course information, not only just a particular point, but also across the timeline. So across the time, where you can integrate all the information to get those key parameters to drive your decision making. So I think that's certainly one thing that we rely on the modeling and the simulation to achieve.
If I could just add to my prior comments, I think there's a tendency for a lot of these more advanced techniques for dose escalation to be tuned in a way that they compete with 3+3 in terms of number of patients used. But nowhere has it been figured out that 3 patient cohorts are sufficient to give any kind of low degree of uncertainty. So I think we need to start revisiting that completely and not tuning these algorithms to say, oh, look, it only uses about the same number of patients as 3+3. Like the stopping rules, I wanted to ask Dr. Conaway, would there be a way to have a stopping rule that's uncertainty based? Once you achieve a certain threshold, then you stop the iteration?
Yeah, absolutely. And we didn't use those, but there are papers out there that try and do, for lack of a word other than the technical one, stochastic curtailment. Here we are today. What's the probability we'll make a decision if we finish the trial? And so always looking at, what would we recommend based on the current data, and how much would that recommendation if we enroll an additional 10, 20, 30, 40. And so there are ways of kind of seeing if it's worth enrolling additional patients for that specific goal. There's always value in enrolling additional patients to get more information about secondary endpoints, about potential subgroups and things like that. But I'd certainly want to endorse what Dr. Patino said about the sample sizes just sometimes being too small, and that the variability that is induced by that just can't be overcome in a sample that small.
Thank you.
Can we just go to the online question and then come to?
Yeah, I have a couple online questions for Dr. Yin. So you note that if there's a PK-DDI in a combination, then the Vikram dose may need to be lowered. So when combining with standard of care chemotherapy agents, can you please share your insights and experience on the extent of evidence needed to select the lowered dose of the backbone agent? And how precisely does the DDI magnitude have to be evaluated? And what are the particular implications in the context of typical cohort size for such a DDI evaluation in a combination phase 1b study?
Yeah, thanks for the question. I think this is a great question. So certainly when we plan for the dose combination, so the DDI, sometimes we have to rely on what we already know from the preclinical stage, right? So based on what we can know from the preclinical assessment, so understand what is the potential for that drug-drug interaction. And then sometimes we use some modeling tool like a SynSIP. This is what we used to predict, for example, in my example, the twofold increment in exposure in drug X because of the combination with drug Y. So that was based on the SynSIP model. But again, the prediction is the prediction. Sometimes you utilize all the best information you have, but in the end, so the data may show otherwise. But this is indeed what we learned. So we need to adjust according to the data. That's what we did. In fact, after we saw the data, we just changed the again, we just updated the model. So taking different assumptions. So that's where, I mean, I think, again, it's going to be a continuous process of gathering all the information to make a decision, and then based on the updated data to make adjustment.
Also, a follow-up, like clarification question is the data on the actual in vivo PK studies didn't show the DDI due to the 3A4 interaction. Is that will that affect your approach?
Yeah. So indeed, in that particular case, so the prediction was based on the fact that there could be a time-dependent inhibitor of CYP3A4. In fact, for the time-dependent inhibition, we know that there's a challenge sometimes in predicting what is going to happen in human. So in that case, indeed, this is what what happened in that the prediction. I believe that there may be some assumptions that we made were not actually the case. So in the end, we were not able to see that the interaction there. But I think again, just based on the need to make certain decisions, we have to make sure that we integrate all the available information and make an adjustment accordingly.
Also, for those one more, for those novel, like novel plus novel combination, was any justification or actual preclinical data that can support this additive effect in your assumptions?
Yeah. Yeah, it's another great question. In fact, I did not mention in my presentation that indeed that was based on some preclinical information that the effect of both compounds on neutrophil was going to be additive. So in fact, we had some PBMC studies done using ex vivo approach, adding the different drug concentration and mapping out the areas where we're going to see overlapping neutropenia effect. And then based on that information, we determined that could be an additive effect.
Thank you. Any other DDI questions?
Thank you. Hi, this is Li Zhu from Bristol Myers Squibb. I appreciate all the very insightful presentations. My question is to the presenters as well as the panelists. I can appreciate the multifaceted nature of trying to do dose optimization for combination. I think doing this for one combination is challenging, but it's not unsurmountable. My question really is maybe the panel can provide some advice or considerations for doing this combination dose optimization multiple times, right? Because a lot of time, our agent has the potential to be used as a backbone therapy. Right? How can we leverage the mono agent, clinical data as well as the initial combination dose optimization, to facilitate subsequent combination dose optimization. I think adding to even more challenging is oftentimes we actually do this type of combination simultaneously, right, in some of the early trials. So any advice and considerations will be appreciated. Thank you.
Maybe I can start.
Sure, please.
So, yeah, certainly from the industry perspective, so we have to be consider the cost and also the timeline as well. So when we do the novel combination, or I mean any other combinations, there could be a myriad of different options to combine the two agents. But we have to prioritize because there's no way we can explore all of them. So then how to make that decision? I think certainly, again, understand what we already know about this combination is very important. So as I mentioned in my presentation, certainly a lot of times the preclinical information can be very helpful to understand the individual contribution. So I mean, in a way to efficiently remove some of the options from, again, that a wide variety of different combinations. And then also the clinical data, early clinical data is extremely important. I mean, given that the monotherapy can tell us a lot of things about the exposure response to both efficacy and safety. So if there's a certain marker specific to a compound, so those can help understand, again, the individual contribution of that agent to a particular response. So that is also very important, again, to remove some of the options from the different combinations. And of course, in the end, we have also to adapt to the data. So during that process, so we have to consider the efficiency. So we cannot, sometimes we have multiple options to test, then we have to prioritize them in a way that will answer the most important question. And also from the clinical perspective, I don't believe that we are at a luxury to answer all the questions all the time. So sometimes we just have to think, what is the most important question for your decision making? So that's, I believe, typically what we have been thinking about as well.
Thank you for your question. So thank for adopting comprehensive answer. So additionally, I think when we propose a combination therapy, we really want to think through what is the justification for such combination, what is the mechanism action behind such combination, and also based on the efficacy and safety and pkpd data from the monotherapy, whether the addition of the drug will combine the drug will increase the efficacy of drug, or add one drug will sensitize the cell and the response to another drug. Also considering the potential mechanism of action for the toxicity, whether it's kind of like a more general overlapping toxicity like a neutropenia or like infection, or we have a specific concern at the pharmacodynamic level. For example, one drug will increase expression level of some like a membrane target on the target cell, which will enhance the efficacy, but may also have a significant increase in safety concern, for example, cytokine release syndrome. Under that case, when you consider the starting dose, we probably would want to be much more conservative to avoid the harm to the patients.
Maybe I can add one thing as well. We can also leverage information from the preclinical model. I think it's also very important because we can also test in vivo what is going on, and then you can leverage from that from a efficacy standpoint. And then what you said, what is very important is to look at the efficacy and if there is some overlapping. So to define the dose at the beginning, and we leverage that from the monotherapy data most of the time.
I totally agree. Thank you.
Please. Several of the presenters gave examples where the drugs or drug didn't act exactly as predicted, which led to the exploration of multiple further dose levels or amendments. And I think one of the presenters also commented that they could have done the whole program faster. Perhaps you could give us some of the lessons you've learned during these programs that would, in retrospect, perhaps have saved you some time or refined your decision making?
I can offer the last part, the easier part about the faster. I can tell you off the camera what went wrong. Many oncology programs early in phase 1, especially if you're not able to do the studies in healthy volunteers in a phase 1 in patients, have kind of startup issues. And in our case, it was a rather unique, unpredictable thing that took us 9 months to enroll the first 2 patients. But it took, and once the floodgates opened, it was clear sailing after that. So some of those things are beyond your control. But certainly, what could we do differently? Having learned the lessons in this case, we're probably a little bit too conservative, being influenced by reports in literature that some patients that are high CDA status patients were amenable to toxicities, and even mortality cases reported with some CDA substrate drugs, not the one that we used here, but gemcitabine, for example. So we were really worried to avoid the situation when we have an extreme outlier would present a different case to manage in the clinic. So maybe we could have started at slightly higher dose of the substrate. But again, the cytidine deaminase is such an interesting enzyme. Not very many people study it, and it's only research-grade reagents. Every lot you get a different data in vitro, very difficult to really feed into your models and expect miracles. So we were very empiric in terms of reacting the evidence as it was emerging from the clinic. To fight for every patient with a PK endpoint. You can imagine making those escalation decisions. I wanted to see at least five or six data points in each cohort. And yeah, I think the next one we can probably cut about a year from the development time.
Anybody else on the panel? Lessons learned?
Can I go? Yeah, so I would say in terms of that one example that I showed that was inspired by a real example, but it was fake data. The real data, I did all this, this fancy optimization modeling, and I was feeling pretty good about myself. And then they said, oh, by the way, the target product profile is 65% response rate. And then, so I reran the graphs, or I ran those probability of hitting the TPP graphs. It was just a sea of dark blue. There was no dose had any probability of doing anything good in reaching that response rate. And so that would have saved me a lot of trouble from a modeling point of view, had I known the basic parameters first. But for whatever reason, it didn't come out till a few weeks into the collaboration.
Okay, next person, please.
Yeah, Azhar Hussain from Merck. So apologize if this question has been asked. I had to step away. So there is a scientific aspect and there is a practical aspect to it, right? When we are looking at the monotherapy combination, we have some exposure response analysis, understanding of your own drug. But there may be case, for example, chemo, the exposure response is not available, right? And for the other agents, they may not be available. When you're trying to combine with an agent from another company, it often becomes a challenge to sort of whether do you have really the dose strengths to manage and develop the combination therapy. Want to hear the panel's thoughts on that. And the second one is, obviously there are cases where it's the chemo dose or other agents dose is practice setting. There is often challenge from investigators to look at another lower dose. From a recruitment point of view, it has been a challenge. Yes, so just want to see where the scientific and the practical reality sort of come to in the middle so that we can take that approach in a proper manner.
Thank you. Just to summarize, the first part of the question is, how do you better use competitor data? The second,
yes, yeah.
And second,
and plus
is,
you may not have the dose strengths.
Exactly. And second is about how do you convince clinicians to change their doses from an already well-established dose to lower the doses?
Yeah, thanks.
It's a challenge. I can speak to the second question first. Once a regimen or a dose level is on the label, it's very, very difficult to convince in research and investigational proposals to do anything different. And I think generating data, whether it be preclinical or model-based data, to convince the agency when you're submitting your protocol that you want to do something different, that would be acceptable. But it's not an easy task, unfortunately.
I'm going to assume this is already on clinicaltrials.gov, so I think I'm okay talking about this. We did that is a difficult proposition when you have a novel added on to either a standard of care or some other marketed drug. We did manage once with alisertib, our aurora kinase inhibitor, when combined with paclitaxel, to have a couple of doses of paclitaxel as well as many doses of our own investigational agent. But the only reason I think at the time, the belief at least was the only reason we got away from that is because paclitaxel has 2 different doses depending on the indication. We just borrowed the lower dose from I think it was breast cancer versus we were studying ovarian. But I remember hearing somewhere that FDA is open to designs that do include non-approved doses of the approved agent. Lower, that is.
Lower.
Yeah.
Maybe, do you want to comment?
I was going to ask a question to everyone. I was going to ask, I guess along these lines, but I know that it sounds like there's difficulty when there's this dose for the standard of care. I guess one option could be a de-escalation type design, particularly if the second agent you're adding on is known to be relatively non-toxic. So what do people think about whether that would be an option, the feasibility?
Can you just summarize your question again? You're talking about de-escalation designs?
Right. So just starting with the standard of care usual dose and then having an option to de-escalate from there initially, maybe 1 or 2 steps. Is that viable or no?
Yes, but it will give you a negative impact.
Could you speak closer to the mic?
Okay, so, but it will give you a negative impact. And when you look at that, you will say, instead of starting with a lower dose and then try to escalate, when you desescalate, it has a negative, I mean, it's more, in fact, on the opinion, I would say that desescalating as a negative is more a negative option than to escalate. So, to my point of view, it's better to try to with a lower dose and try to escalate. Yes, than the contrary.
So another concern is that worry about one of the concern we is like the patient cannot tolerate the higher dose and have to have multiple step dose de-escalation, so which maybe it will do more harm to the patient. Maybe just as Doris just mentioned, it's better to find a dose that just be good fun. It's a good dose from the beginning rather than de-escalate.
In fact, I just want to maybe address the second question. So that is about the investigator's hesitation to test a lower dose. Sometimes that is believed probably less efficacious. So indeed, I think this is a challenge that we face. In fact, in one of our programs, so we had that challenge from the investigators that, well, they don't believe the lower dose is going to be as efficacious as the dose that is already very well tolerated. So in fact, I would just be very interested to get the maybe perspective from our colleagues in the regulatory agency. How do you look at this? So sometimes we know that a regimen is well tolerated. I mean, they're efficacious. And the investigators, they're hesitating. I know that one of the suggestions from the agency is to really look at the randomized approach, but still that's going to add a lot of patients unnecessarily to test a dose that is not necessarily optimal for the patients, which we may already know. So again, just maybe for our colleagues in the agency to comment.
So, Dr. your question is that you have a dose hypothetic drug that is you consider is very well tolerated, and you want to try a lower dose, which then hesitancy from the investigator. So.
Yeah, that's right. I mean, just maybe more specifically in that case for our drug Y. If a drug Y is not, I mean, what I mentioned that in the example, so we have the dose level 3 and dose level 4. And then the lower doses, we know that probably is not going to be, again, from the target coverage perspective, it's not going to have the same target coverage. And also dose level 3 and 4, they're very well tolerated. But still, I mean, according to the guidance, we have to comparatively assess the lower dose compared to the dose level 3 for optimization.
Okay, so for this kind of like very specific question, probably as a sponsor meeting with more open data will be more helpful for both of us.
Yes, thank you.
Yeah, I think that's it'd be kind of a rare situation that you would know that much about your drug after a phase 1. I mean, I guess you'd be very lucky to know that both these doses are extremely well tolerated. Usually, you know, the spirit of the randomized dose cohort is that you don't really know that much about either one of these doses. You need to learn a little bit more, both in terms of efficacy and toxicity. So I take your point, though. If you already know a lot, do you need to further optimize? I think that is, I don't know, is that type C meeting? I don't know.
I guess for one or two very specific questions, that will be type D meeting. Also, you just mentioned the drug is very well tolerated. Then probably that's from your perspective, and we would like to see the data and based on our knowledge for the overall landscaping to evaluate whether that holds true from our perspective.
There is a type meeting going on here right now. So
thank you. Yeah, thank you.
Let's move on to the next question here, please.
I don't have a question. I just had a comment from my clinical perspective at FDA. I think the original question that got asked was, are we open to evaluating dosages that are different than the approved dosage? And the answer is yes, with appropriate justification. You know, that's why I said in my talk earlier today that an important consideration is, have the dosages been optimized for the individual agents, even if they're approved as monotherapies? Additionally, there might be data that emerge from the post-marketing setting that's not necessarily, you know, a significant safety signal for a serious or life-threatening event, but that the drug is being modified, the dosage a lot by patients, or is more poorly tolerated than thought. So yeah, we are open to that with sufficient justification. I also think just the comment on patients and investigators being resistant to a lower dosage, definitely there are patients and providers that have been telling us for a long time, we want lower dosages, different dosages to be evaluated specifically to look at tolerability. But there are other, you know, there are a mixture of attitudes in every stakeholder group. And I do think some of these attitudes might change with time. But just 2 comments, thanks.
Thank you.
There's this phenomenon I'm sure it happens at every pharmaceutical company, but at Takeda we call it dissent by proxy, which is it's the easiest way to squash an innovative idea is to say, I like your idea, but the FDA will never go for it. Or I like your idea, the clinicians will never go for it, or whatever it is. But it's important to test that out and not to take someone else's word for what a third party not in the room would say about your trial design.
They tell us that all the time as well.
We'll take one virtual question and then come to you, please.
Yeah, I have one more online question. For those combination for novel modalities, for example, ADC or bispecific, what is our dose optimization concerns? For example, the ADC is with lower payloads, and the bispecific combined with bispecific or other novel modalities. What is our thoughts?
Okay, I think I can start the question with the bispecifics. So for the bispecific dose optimization, so we generally consider like 2 parts. One is during the initial part, that's a step-up dosing that probably try to mitigate the cytokine release syndrome, and the later part will more focus on the longer treatment dose, will be more like efficacy and long-term safety signal. So for the in combination with another agent, we want to understand what is the potential mechanism for this add-on therapy. For example, if that is just mentioned earlier, if an add-on drug such as gamma secrets inhibitor that will increase membrane BCMA level, then if this drug is added on to the early part, then we have a concern on the cytokine release syndrome, the safety signal. So we probably will figure out how to adjust the starting dose, re-evaluate the starting dose, the step-up dose for the bispecific antibodies. Then for the therapeutic, then for the later longer phase, the therapeutic dose level, then our concern probably will be overlapping toxicity like cytopenia, infection of the bispecific and the combination agent. So under that kind of condition, probably we will try also evaluate whether a lower dose or less frequent dose can be equally effective, but have the improved safety profile, and that can for both early phase step-up dosing and the later phase treatment dose. We would recommend use of quantitative assessment, the PK analysis, and probably more involvement of mechanistic modeling, because bispecific have unique characteristic, which is driven by the interaction between the drug and the T cell and target cells. You want to make sure you do get a detrimental dose escalation, both from the efficacy and the safety perspective. Then for the ADC, sometimes you will also think about the interaction between the ADC, the antibody, as well as the payload, for whether there will be kind of interaction at both PK and PD level that may affect the efficacy and the safety, and you rely on the totality of the data to make the decision.
Mark Retain, University of Chicago. I want to add to Dr. Shah's comments about lower doses of a partner drug. Not only is FDA receptive, they've done it. And so you could look at the original approval of Tykerb, where Lapatinib was approved in combination with capecitabine at a lower dose than was in the Xeloda label. And so that's been done. The other point is, in the context of Project Optimus, I think it would be in sponsors' interests to use a lower dose than the label dose if the label dose was never optimized, both to reduce toxicities of the combination, but also to improve cost effectiveness of the combinations. You're adding a new drug that's going to have costs. Well, why not decrease the cost of the other drug if it's not yours? So that's something else that sponsors should consider.
Thank you. Jen? Sorry, are there any comments? All right.
Jen Genetech, this is a general question for the panel. All right, I shouldn't have touched that, sorry.
You're supposed to make your hard question and then drop the mic.
Okay. Anyway, at the time of expansion, a common dilemma we face in drug development is there are so many things we want to test. You want to test multiple cancer types. You want to test particular mutation subtype of patients. You want to test different combinations, and you want to test different dose levels. So how to prioritize? Do you prioritize one more dose level? Do you prioritize testing 30 patients rather than 20 patients, or testing multiple dose levels in both in multiple cancer types or combo settings, or testing additional patients and populations? I'm curious what's the panelist experience and insight on this, especially from today's typical practice in drug development and regulatory perspective as well.
It's open for everyone.
Okay, I think maybe I can have the first shot. So, from our current practice, we would, during the dose escalation, especially for solid tumor, we have the solid tumor, the dose escalation conducted in all comer. But then during the dose escalation, when you find some of the cancer subtype with efficacy signal, then you may consider to conduct dose expansion in those cancer subtype with a signal, and in those subtypes probably expand to more than one dose. When you talk about the patient number, I understand we always say a sufficient number of patients, which probably will be justified by the rarity of the cancer and the information and the leverage information from both or probably from all the data to help you inform your decision of RP2D selection.
I can try to speak from like a project team representative at the company. So to keep the interest in the program going, obviously, I would think you would want to prioritize the signal finding to get enough activity to justify additional. So you can just rank order your spending, how much money you have. If there is money available to show that your initial hypothesis, whichever mechanism you were targeting, is generating signal activity sufficient enough to justify further investigations. A very good, safe dose, well-tolerated drug without sufficient activity, very difficult even to convince to do even a combination trial next. So even combination products are expected to have some sufficient good enough activity. So for prioritization in phase 1B, probably that would be my recommendation.
I think that's exactly why at that time, generally in the cross-functional discussion or debate, a lot of time it's really prioritizing the cancer types that we may more likely to see a signal rather than, so therefore we will generally as clinical pharmacologists, we struggle to argue for testing that one dose level. We'll be lucky if we can do 2 dose level in one indication. If we want to do that in multiple indications, even if there are potential differences or different combinations, I don't think that's a battle we always win. So I'm curious for the rank order prioritization, especially from other industry colleagues.
Yeah, so I can just add to what has been discussed. I think certainly, again, the prioritization maybe start with the prioritization of the question. I mean, what is the key question we want to answer, right? So is that which indication we want to pursue at the pivotal trial? And also for the different regimens, so what are the variables that we need to understand? I mean, those variables. And then based on those prioritization of the questions, we arrange the corresponding experiment to get the answer. So again, a lot of the times, so we have to probably just deprioritize or not be feasible to test all the different options. So we then identify the key factors. And then once, another approach I would say that, well, is to rely as much as possible about the extrapolation of information. I mean, what we have learned from the prior clinical experience, or I mean, when we think about a particular experiment to do, hopefully the information get from that particular setting can be applied to multiple indications or multiple combinations. So that is where probably we can get the information more efficiently. That's how we can look at this as well. Yeah, again, I agree that this is a very realistic challenge in that, again, so many questions we need to answer. There's no way we are not feasible as well to explore all the different experiments.
Thank you.
Thank you. Yes, we have time for one last question before we close the session.
Yeah, thank you for the speakers and panelists. Benjamin from Adagene. My question is on the practical side of setting the optimal criteria or threshold for safety and efficacy, particularly given that the same combination could have different types of criteria depending on the medical need, right? An unmet medical need and also emerging therapeutic combination that's really generating new results. So I would like to understand a little bit more about how important, right, is it to set that criteria and at what stage during the drug development? Thank you. Yeah, for example, you can address probably just on the safety side. Same combination, different cancer type, but the unmet medical need could generate different needs for reaching potentially different threshold.
It looks like an extension to the 65% TPP that Dr. Bottino was talking about.
Okay, yeah, I can take a first crack or a crack at it. What's he gonna say? Well, I lost it for now. I'll be back.
So just to clarify, this is Jonathan. You're talking about setting maybe like a target DLT rate in a phase 1, or you're talking about something later in
later, for example, like phase 2 setting, phase 3 type of doses, right? Like, is it acceptable to have different types of, let's say, DLT rate or other long-term safety percentages, right? When you're thinking about different cancer types?
Well, I would think so, but it'd be hard for me to tell you what they are. I think it gets at something that I've been thinking a lot about that I'd be interested to see if anyone has thoughts on this. We're talking a lot about in phase 1, we're only using the DLT window as like 1 cycle typically. And so our target rate there is 25% typically, or 20. Well, if it's 2 cycles, should it still be 25%? If it's 3 cycles, now you're asking long-term till progression, that might be 6 cycles, that might be like 20 cycles, depending on what the drug is. So what is a reasonable cumulative rate there? That I'm not sure. I mean, I do think that that's something, I wonder if there's research out there on this, but I do think that is like a worthy area of research, is to kind of understand, okay, what can we expect and what is tolerable and what is likely to inhibit efficacy. And so I don't know, do you have any thoughts, Dr. Conaway, or anyone else here who's in this area?
No, I think that's an excellent point. I mean, and that kind of goes back to, I think your question is about tailoring the design to the objective, set the threshold based on what it is, the particular clinical question of particular agents. So I think, yes, there is some room for moving those thresholds around. And I think also there has been some work, I'm blanking on the exact reference, that look at how the phase 1 thresholds and phase 1 experience predict later, phase 3 later. And I think that's, yes, an important area to be looking at is updating that work to how does what you find in the phase 1 predict what you might see in like a registration phase 3 trial.
Dr. Ajay?
I have to respond as a physician, okay? The target rate is 0, okay? In the greatest health emergency of our lifetime, how many trials of COVID, anti-COVID drugs were done with a target dose-limiting toxicity rate? 0. So we should stop talking about MTD and target DLT rate and stop designing trials that way. It makes no sense medically, and it certainly makes no sense to patients.
Yeah, to that point, and that reminded me of what I wanted to say before. I have never seen a non-inferiority study in oncology where you would have non-inferior efficacy but a better safety profile. But are there examples out there of that?
I think there should be, but I don't like non-inferiority either as a study design. And I'm collaborating with someone who feels very strongly about this and has published on this, Charles Mansky, who's an economist at Northwestern, who's an expert in decision theory, econometrics, and medical decision making. He just had a PNAS paper about this, and he's got a NBER paper out there. And he's a patient himself, and he published his story in the New England Journal early this year. Patients don't want to hear about toxicity. And it's fine. A label can have a range of doses, and if the higher dose has marginally more efficacy and clearly more toxicity, the prescriber and the patients can decide what dose to utilize. But there's just the notion that we have a target dose-limiting toxicity rate. That's a constraint. That's not a target.
With that note, I think we'll end the session. Thank you, everyone. We will reconvene at 2 p.m. So the lunch is right outside, actually, across the hall. Thank you, everyone, and thank you, speakers.